Horizontal gene transfer is a genetic mechanism in which an organism is able to directly distribute certain multiple gene segments of its DNA to other organisms that are around it without even undergoing reproduction in the first place. It was recently thought by the scientific community to have more significance in prokaryotic species like Bacteria and Archea than eukaryotic animal species. Horizontal gene transfer is very common in antibiotic-resistant bacteria. When a bacterium becomes resistant to an antibiotic drug molecule, it often tends to share the same resistant gene directly to other bacteria that exist around it, causing many bacteria in the same population to withstand against the drug molecule. Because of the vast significance of horizontal gene transfer in prokaryotic microorganisms, evolutionists thought that it was common only at the bacterial level of prokaryotic species and not so much in higher microorganisms. Given the lack of horizontal gene transfer in higher organisms like humans, cats, dogs, and so on, it is safe to say that the genes observed in higher taxonomy can be used to construct molecular phylogenetic trees to establish which species are more closely related and which ones share a common ancestor. Since genes in higher taxonomy cannot be horizontally exchanged directly, unlike bacteria, it is safe to say that those genes were inherited through the lines of ancestry from one generation to the next rather than being directly exchanged. In addition, if by any chance horizontal gene transfer was indeed found to be common in higher organisms, our hope for constructing phylogenetic trees will be in serious trouble because it will show genes independently, separately, overlapping in other species' DNA without even passed on vertically through reproduction from one generation to the next. This is extremely unlikely, and so there is nothing to worry about, or so they thought. In the article from the prestigious scientific website The Scientist, titled DNA Jumps Between Vertebrates, researchers have found a retrotransposable genetic DNA segment that can copy-paste itself in a genome named BOVB. BOVB gene is reported to be common in cows, but surprisingly, they have found the same gene to converge independently among different organisms as quoted below. BOVB is a retrotransposon, a piece of DNA that can copy and paste itself around the genome to create large sequences of repetitive DNA. It is abundant in the cow genome. Researchers have also found it in the DNA of many other animals, including elephants, horses, platypuses, pythons, sea snakes, geckos, sea urchins, and even zebrafish. Interestingly, they found another unexpected discovery. To better understand how BOVB found its way into such a diverse range of creatures, researchers used sequences from these animals to construct a phylogenetic tree. If BOVB has been passed down from a common ancestor and remain in the genomes as they diversify, then the more closely related species will have more similar versions of the DNA sequence. But that was not the case. Instead, the team found that the BOVB sequences from cows were more closely related to snakes than elephants. The researchers suggest that the only explanation for such unexpected evolutionary relationships is that BOVB has jumped between genomes, and they figure that it must have done so at least 9 times. Finally, the researchers identified a potential vector. BOVB sequences were found in 2 tick species that suck the blood of lizards and snakes, indicating that the parasitic arthropods could be responsible for passing the jumping genes between species. Was this? Apparently, the phylogenetic tree has failed to construct the tree of life of the BOVB gene, being similar to closely related species as commonly believed by neo-Darwinian evolution. Instead, we have a phylogenetic tree that does not fit with the expectation of evolution, since the molecular phylogeny of BOVB gene has become too corrupted to establish common ancestry relationships, given that the gene has jumped horizontally and directly from genome to genome in a broad range of eukaryotic species. Furthermore, Scientific American has published another similar article with the quote, "Multiple independent gene transfers are now documented to occur in the evolutionary history of eukaryotic life, not just among prokaryotes." Researchers did a cross-analysis of the bacterial gene ACDS from the genomes of 149 different eukaryotic species and found the same bacterial gene exists in 65 different eukaryotic genomes. Muller and his colleagues scanned the genomes of 149 eukaryotes and found ACDS-like genes in 65 of them, 61 in fungi and 4 in parasitic microorganisms. After analyzing the organism's genetic family trees, the researchers determined that the most likely explanation was that 3 different kinds of bacterium had donated the gene to the fungi, in a total of 15 different horizontal gene transfer events. Because of the increasing evidence of horizontal gene transfer becoming common in eukaryotes and not just in prokaryotic species like bacteria, the phylogenetic tree to establish common descent has become rather elusive and quite difficult. Charles Davis, an evolutionary biologist at Harvard University in Cambridge, Massachusetts, says that the work shows that the commonality of gene transfer between bacteria and eukaryotes is unexpectedly high, but it faces the same problem as many such studies. The analysis of the genetic trees is not detailed enough to clarify where and when the transfer took place. Ironically, genome biology researchers have commented to the issue of horizontal gene transfer and how it challenges phylogenetic trees, as referenced in a scientific article titled "Some Genes Foraging in Origin and Not from Our Ancestors," and quoted below. Lead author Chris from the University of Cambridge, United Kingdom, said, "This is the first study to show how widely horizontal gene transfer occurs in animals, including humans, giving rise to tens or hundreds of active foraging genes. Surprisingly, far from being a rare occurrence, it appears that horizontal gene transfer has contributed to the evolution of many, perhaps all, animals, and that the process is ongoing, meaning that we need to reevaluate how we think about evolution." The researchers studied the genomes of 12 species of fruit flies, 4 species of nematode worms, and 10 species of primate, including humans. They calculated how well each of their genes aligns to similar genes in other species to estimate how likely they were to be foraging by origin. By comparing with other groups of species, they were able to estimate how long ago the genes were likely to have been acquired. A number of genes, including the ABO blood group gene, were confirmed as having been acquired by vertebrates through horizontal gene transfer. The majority of the other genes were related to enzymes involving metabolism. In humans, they confirmed 17 previously reported genes acquired from horizontal gene transfer and identified 128 additional foraging genes in the human genome that have not previously been reported. The author says that their analysis probably underestimates the true extent of horizontal gene transfer in animals, and that direct horizontal gene transfer between complex multicellular organisms is also possible, and already known in some host-parasite relationships. Ouch. So apparently, as it turns out, there are genes in the genomes of higher eukaryotic taxon that were transferred horizontally rather than from a common ancestor, as was commonly believed. Horizontal gene transfer could explain a lot of why many species tend to have many different types of genes from other different types of organisms, including the eukaryotic ones. For example, the famous claim that chickens have the genes for teeth. It could be that instead of those genes originated through ancestry as evolutionists believe, those genes were rather transferred horizontally, independently, and separately that had nothing to do with the inheritance of common descent. Upon having this video published earlier, I stumbled upon a criticism in which I think deserved immediate attention. The author of this criticism was his YouTube user and defender of evolution, Rational Mind. Mind claimed that genes as complex or similar as the chicken's teeth cannot be horizontally transferred because apparently it is too complex considering the regulatory and developmental aspects. By contrast, he explained that the reason why lateral gene transfer was able to transport the ABO blood gene instead was because it was less complex than the chicken's gene teeth. Unfortunately, what Rational Mind failed to take notice was that horizontal gene transfer can too transfer genes involved in regulatory controls. One paper titled "Horizontal Gene Transfer Beyond Genes," Eugene Kuhne described what is known as horizontal regulatory transfer. It is where the non-coding regulatory regions of DNA undergo lateral gene transfer. Its purpose is to modify regulatory switches and even rewire the overall regulatory system of a given biological organism. A genome analysis of 46 sequence isolates of E. coli reveals that the majority cases of regulatory regions were the result of horizontal gene transfer transporting the regions along with that of its downstream regulated gene. By contrast, only 11% of the regulatory regions evolve in the transfer of those regions alone without transferring the downstream regulated genes. In other words, the majority of the regulatory regions involve the transfer of regulatory controls together with that of genes corresponding to the regulatory region. Unfortunately, as expected, if Darwin's tree of life was false, the paper concluded no phylogenetic evidence in favor of the tree of life model since it generated numerous conflicting species trees. So, if horizontal gene transfer can not only just transfer active genes but regulatory ones as well, including the genes undergoing such regulation, then I don't see why something as complex as a chicken's teeth that involve regulatory developmental pathways cannot be done. Theoretically, it may be possible too that the regulatory regions found in eukaryotes could be explained by horizontal gene transfer. I suspect this to be the case as more papers are published. Now, putting the devil's advocates even further, I stumble upon another interesting paper. Here, the authors describe a eukaryotic gene named EFL, elongation factor that is present only in eukaryotic genomes. This gene was widely believed to have originated vertically, that was inherited from the last universal common ancestor. However, as the authors have implied, large-scale sequence data from the phylogenetically diverged organisms challenged this assumption. Instead, phylogenetic analysis described strongly that lateral transfer was the cause of the EFL gene being present in eukaryotic genomes and was not the result of that gene being passed down vertically from a so-called universal common ancestor. I guess it isn't far-fetched to suggest that genes can jump too from eukaryote to eukaryote genomes. Thanks for watching.